So today I'm going to be talking about establishing high-throughput phenotyping methodology in a sorghum breeding program. So sorghum, it originates from Africa. In the United States, it is traditionally grown as a hybrid crop, with some of the main uses being animal feed, food for people, so like cereal, and then it's also as a bioenergy crop. In 2020, 5.8 million acres were planted in the United States, with 1.8 million acres being planted in Texas specifically. We have a sorghum breeding program here, as you guys know, and a few of the things that we are working on is disease-resistant varieties, specialty hybrids, and most importantly, increasing sorghum yields. So over the past few years, we have been working on increasing yields through using the modern-day breeding triangle. Modern-day breeding triangle includes genomics, phenomics, and environomics. Genomics is genomic prediction models, as you heard last week from Noah. Environomics is including environmental data to predict your hybrid performance. And then phenomics is stuff like your UAVs. And I'm going to be specifically talking about UAVs today. All three of these things work together they can be used to predict hybrids and their performance in the field. The main objective of this talk is about how we found a UAS for our breeding program here, and then how we determine the optimal height to fly the UAS to collect vegetative indices. And then finally, I'm going to talk a little bit about how we assessed vegetative indices collected in the morning and afternoon to further define limitations of those indices. So starting off, first, what unoccupied aerial system should be used? We were faced with this exact question about 2 years ago. Looking at research, a lot of people use DJIs, and that's just a brand of drone. And then a lot of other people are building their own drones. So they'll go out, find a specific camera they want or a specific drone and put them together for whatever they're researching. There's a lot of different UAVs and cameras on the market. A few of the most common UAVs are fixed-winged helicopters and multi-rotors. So fixed wings, these are great because they can cover a lot of space very quickly, but they do need a landing strip. So if you're out in the middle of the field, no roads around, you're going to have issues landing. And then helicopters, these are also good because they have a high payload capacity, so you can put extra cameras on them. But there's an issue since they're a little more complex to operate than other drones. And then finally, we have multi-rotor, which these are really good because they can hover. And so that means you could just stay flying right over one plot and get a lot of images over it instead of flying quickly like a fixed wing. And usually they're very affordable. For a few of the common camera types, there are RGB, multi-spectral, hyperspectral, and thermal. For the RGB, that's just covering the red, green, and blue wavelengths. Multi-spectral, it's the same as RGB, but usually it also covers near infrared. Hyperspectral is when you're going to be able to cover a lot more bands over the spectrum, so it's a lot more data points you're collecting. And then thermal, that's what you're using to measure temperature with long-wave infrared. There's a few other issues that we have to pay attention to at A&M instead of just deciding between, quickly common UAS and camera on the market, and that's that at A&M we cannot purchase DJI or foreign-made drones anymore. And as I previously said, a lot of research is done with DJIs. So that'd be pretty easy for us to instantly get a DJI, start reading research papers, just replicate stuff, and start phenotyping stuff very quickly. But since we cannot purchase those due to the fact they're not NDAA compliant, basically it's just the United States saying, we're scared these foreign-made drones are taking our data, we had to look at other drones on the market. And we decided to go with the Skydio X2E. This is a multi-rotor drone. It has an RGB and thermal camera on it, and it also has a 35-minute flight time for each battery. It also has 360 obstacle avoidance. So that basically means that if we're flying close to something like the ground or a tree, it's going to stop and hover and it won't crash into the object. That's great for when you're trying to train someone new how to fly. It's less likely to crash and you won't have to replace it. It's also NDAA compliant, so the school's okay with it, but there is one big issue with that, and it's that it's a newer drone on the market. So all the software that's currently available to process imagery and plan flights, it's not available yet for this drone. And so we had to find other methods to process the imagery and plan all those flights. So after we got the drone, we were unsure what altitude to be flying at. And that's because there's really no clear answer in research. A lot of research doesn't even address what altitude they flew at to collect data. And then other research is only doing it based off what thing they were studying. So there's research on panicle counts, but that's done at 10 meters. And then other people have done research on vegetative indices, but that's done at 60. We wanted to be able to go out in the field and collect all this information at once instead of doing, 10, 20 flights to collect every single different trait individually. So what we did is we flew a hybrid grain sorghum field in College Station last summer. This field was made up of recombinant inbred lines crossed with 4 common male testers. So we had a total of 802 row plots. And you can see right here that there's good variation in the yields and plant height. So it wasn't just one common hybrid. There's lots of variation we could be looking at through the study. And there's also 5 GCPs in the field, which were later used to georeference all of our orthomosaics. For the flights, we flew over 3 different days, all at solar noon and an altitude of 15 meters, 30 meters, and 45 meters. So we ended up having a total of 9 flights across all those days and then all those altitudes on each day. All of these flights were done at the end of the growing season, and we did those flights then because there was previous students here who also had worked with UAVs in our group, and they were showing that around the area that we flew, that there's also high correlation to plant health and yield. Throughout the 3 days that we flew, the weather was very different. So on day 1, we did have overcast weather. Day 2, it was slightly cloudy. And then day 3, it was a clear sky. For processing all the imagery, we went through and first created orthomosaics in AggieSoft Metashape. Then in RStudio with UAS tools, we created shapefiles. All those shapefiles had a small plot buffer just so we weren't getting the edges of the plots. Edits were done to the shapefiles in QGIS. And then vegetative indices were extracted in Field Image R, which is a package in RStudio. And then finally, analysis was done also in RStudio. So for all the vegetative indices, we ended up extracting 24 of them in total. Why should you care about these? Well, they're a spectral measurement. So basically, it's more phenomic data for our program. A lot of them have been found to be highly correlated to yield, biomass, vegetative health, and there's a lot more studies coming out about why they're good. I'm specifically only going to be talking about VARY, which is visible atmospheric resistance, just so don't have to go over all 24 today. And you can see that this is the formula, green minus red over green plus red minus blue. That's the wavelengths. And it's also been found to be correlated to vegetative health yield. So it's an important wavelength or indice. So straight to the results for the different days and their impact on flights, or the altitude across days and their impact on flights. You can see here we have on the x-axis the days, and then vary on the y-axis. The white boxes, those are the vary from 15 meters, the grays from 30 meters, and then the dark grays from 45 meters. You can see that across days, those means are changing, and then also across distribution, the altitudes. We can't exactly say that it's due to the altitude that those distributions are changing. It could be, but you have to remember we also had different weather on each of those days. We looked a little further into this by running a mixed model. It included altitude, day, and genotype, and then rep was random. The first thing I want you to look at is altitude by day by genotype. This is not significant. So that means across all the days and altitudes, the genotypes were not changing ranking for vary. And since this is not significant, we can also look at the second order interaction, altitude by day by genotype. So again, we're very interested in seeing if you fly at different altitudes, is the order of those genotypes changing? And it isn't. And this applied for all 24 indices we looked at. And so since the ranking is not changing, we can look at other factors that influence our decision on what altitude we want to fly at. We decided that 15 meters wasn't going to work, and that's just because computationally it was way too heavy. So it took a really long time to create orthomosaics, extract indices, and we just couldn't put that much time into waiting for it to run on the computer. And also the flight times were very long. So we flew over 800 plots. If we were to increase that much more, we're going to have issues with needing to buy more batteries for the drone. And then 30 meters, you can see here, that was very good because it had a lot of very consistent images where you could see everything in the image, like the panicles. So if we were to do any objects-based model classification, that's what those images would be great for in the future, because we can easily train the models and then use them to classify things like panicles. And then 45 meters, this one we also really liked, and that's because there was a very fast flight time, which everybody likes, because then it's less time out there having to fly the drone, and then also less batteries so you can use in one field. So you can go to more fields that same day. And it also gave good consistent vegetative indices, as we saw from the combined analysis. So in the future, we'll be using the 45 meter for if we just need vegetative indices, and then anything else will be at 30 meter. So for our last study, we looked at, does the time of day significantly impact vegetative indices? And that's because we have trials across all of Texas, going into Kansas, and we want to be able to collect information from all of these places. But of course, when we drove to these places, or even here to the farm in College Station, it was usually raining at noon or too windy, so we couldn't fly. So we ended up looking into current research about changing that time from flying at noon to other times. And some of the research was saying that you should fly at noon to stop the backscattering of light, which might be affecting vegetative indices. But then other research was showing that hotspots will occur in the field. So these hotspots will actually throw off your vegetative indices at noon. So maybe you should look at another time when these spots aren't occurring. We use the same exact field as we did in the last study. It had the 800 two-row plots, the 5 GCPs, but this time we flew on 3 days, in the morning and at solar noon at 30 meters. So we had a total of 6 flights. And solar noon is basically the time of day when the sun's straight up in the air. So here it's not at noon, actually. It's actually at 1:30 in the afternoon. So we were flying around 1:30. And then in the morning, that was at around 8:30 in the morning. And then all of these also, again, was the 24 indices. I'm only talking about vary. So here's the correlation of vary in the morning and at solar noon. You can see it's a pretty good correlation. It's 0.7, and that's from day 1. And then day 2 and day 3, they were also both 0.8. For all of the indices across comparing morning to solar noon, it averaged out to be 0.68. So they were correlated. Not super high correlation, but they are slightly correlated. Now looking at the distribution of vary across days and times, you can see again, days on the x-axis and varies on the y-axis. The white boxes are from the morning and the dark grays from solar noon. Again, the means are changing across day for all of these flights, and the morning ones are usually slightly higher. I can't say again if it's due to the morning and solar noon specifically. It might be due to the weather. On day one, the morning flight, there was dew on the plants, where in the other mornings there wasn't. So that might be affecting those averages. For the combined analysis, we again did a mixed model with being random and then included the factors time, day, and genotype. And time by day by genotype was not significant. So again, our rankings are not changing between the morning and the afternoon flights for the vegetative and C vary. And then looking at the second order interaction, time by day is not changing, or time by genotype is not changing. So that's just showing us, those rankings are not changing, and all the other indices followed this same pattern. You can also see that time by day, or day by genotype, was changing. That was significant. We're still unsure completely why that's happening, but there are possible corrections we could put in place. Some other groups are trying to put corrections on using environmental factors to correct for that. But it's something just to keep in mind that different days, they might be changing for your vegetative indice. So in the future, we will be flying in the morning if need be. But there are some things that need to be considered, and that's, again, we're using rankings. We're not using exact values from these indices. And that's because those values are changing, and we just want to see what's the highest for that each indice and what's the lowest ranking genotype. And then also, as you would expect, the morning flight, you're seeing more shadows. So if you're wanting to use the morning flights to collect imagery for object-based classification, those shadows will be messing up your imagery. You could extract them or build them into your model for object-based classification, but it's going to be a lot of extra work, so you might as well just wait to fly at solar noon if you can. And then finally, multi-spectral, it will be different, and that's because you're going to be needing to apply atmospheric corrections to your camera. It's someone else. We don't have multi-spectral. You'll have to try this on your own camera. But multi-spectral are usually a little more touchy to all the changing light and stuff. So in conclusion, we will be using an altitude of 45 meters for all of our flights, unless we are wanting to build any object-based classification models. As I just mentioned, the time of day does not matter, but we still will be preferencing noon unless we know that it's going to be raining every day for the next week at noon or way too windy for us to fly. And then most importantly, we now have a UAS in place to use in a pipeline to start collecting phenomic data for our program. In the future, we will be replicating this study to extract heights from all of these images that we collected. And so we can see once again, across different altitudes and time of day, will the height be changing that we extract from all the plots. And then we will also be combining our phenomic data with genomic data to create predictions on hybrid performance. So with that, I would like to thank my graduate committee, Dr. Rooney, Dr. Murray, and Dr. Pescu, and everybody in the Sorghum lab, the Corn lab, for helping me start processing all the data and how we should do that, and then Texas A&M faculty and staff and Skydio. With that, any questions?
Questions?
Great presentation. I was just wondering, you said you used ground control points for each. Were they permanent ground control points, or did you set them out every time that you flew?
So I set them out every time. However, there was bricks left out in the field that had flags around them, so I knew exactly where to set them. And I also took those GCP measurements multiple times. I think it was like a total of 50 times for each point. So I did count for some of that slight movement.
You stand by. I want to change the batteries in the microphone. Yeah, you can do the chat. Read the question out.
Okay, could radiometric calibration of images for each flight allow to compare the indice values between the time of day? It should be able to, but again, it's just another thing that we're going to have to test. Other groups are looking into it along with the combining environmental data. It's just another study and another thing we'd have to apply to this. In the future, we'll look into it so we can combine more data across locations. And then the second one, what do the vegetative indices tell you? Does it allow you to rank each cultivar based on yield, or does it indicate plant health? So the vegetative indices, right now, a lot of people have been looking into them because they're correlated to things like plant health, yield. Some people are trying to find them correlate to specific things in plant health, but it's very specific. Each one's different. Some of them are just looking at soil. So it depends on the indice.
Other questions?
You mentioned predicting hybrid yield. Are you planning to do it with the particle count and integrating it with genomics? Is that the plan?
First, we'd like to try it with just vegetative indices, since they predict a lot of different things for the plants and the environment. And then eventually building a model to count panicles. Yeah.
So out of curiosity, how do you know the indices that you're taking are related to, say, ground truth? What methods do you use to verify these data?
That's a good question. A lot of people have been looking into it. It's kind of like, what is truly the ground truth? It's more of correlations, really. You don't know exactly that's what you're measuring. That's why when you're looking at things like plant health with indices, you can't say a specific disease is causing it, like that, what you're seeing in the indice. And that's because these indices are just looking at like the light that's being reflected back from the plant. So just because you're trying to measure one disease doesn't mean that another one, environmental factor or another disease on the plant, is interacting with them. So it's kind of like, it's just more of a correlation at this point. Yeah.
So you decided on 45 meters as your flying height, but you noticed that it struggled with object-based classifications. Do you think if you applied like a high-pass filter at that 45, where it would kind of get that edge effect more, do you think that would help your models?
Of that orthomosaics, they're created through stitching hundreds of images together. So when you're stitching those images together and they're really far up in the sky, it's very difficult to see the panicle. And then when they're stitched together, it kind of just looks like a blur. And so when you're trying to train a model off of it by circling that panicle, you're not exactly sure if it's a panicle, if it's a leaf that's just brown. It's difficult to tell. When you're closer, like 30 meters or even 15 meters, it's a lot easier for those models to stitch the images together. And you can for sure say, yeah, that's a panicle. I can circle it, train my model off of it.
So at the time of year that you were doing that, taking these measurements, you said there was a variation in the over days. How many days was that? Like 3 days apart?
It was a total of 7.
So how many leaves is sorghum adding a day during that time period?
Oh, it was already, I mean, it was close to being ready to harvest it. So,
So it would have been going
It
through
was-
senescence and
Yeah.
anything.
yeah.
Thank you.
Very quickly, when you think of what we're doing with genomics now, we don't care where the markers are. We don't care whether they're linked. It's just they're added to a model. So do you see the future of phenomics going to where you're targeting phenotypes you think are correlated with yield to augment your prediction models? Or are we going to get to the point where you're throwing like NIRS data at it and you can't say why it would be related to yield, but it's improving your model?
I think the first step will be predicting, if you're looking at yield, predicting what your yield's going to be before you harvest, but your crop's still out in the field. Or going out there with like biomass, sometimes we can't get out in the field and harvest biomass before it's flat on the ground. So going out there and being able to predict things like that, and then eventually just adding in rankings of these indices into our models. If that answers.
I guess all I'm asking is, will we get to the point where we're just in sort of looking at phenotypes that we don't think they're related to grain yield, but they may help your prediction models? Just kind of like markers, we don't know they're linked to a specific QTL. It's just you use all the markers you can, and whatever improves your model, that's what you use. That's kind of what I'm asking.
Yeah.
What I see here is you're targeting things that you believe are related to yield. Will we get to the point to augment our models for things we don't know are related to yield?
I think that's how it will go, because a lot of these vegetative indices, it's just correlations that we're finding. So at the end of the day, some of them we have a stronger belief that they are truly related to yield because they're more related to photosynthesis, something like that. But the other ones are like, well, they're correlated, they'll help, but we don't know how they're correlated yet. So, and it kind of goes back to the ground truth data. We don't have that for a lot of our stuff. So.
Thank our speaker.